Hello, everyone, and welcome to the second plenary lecture of this year's North American CF Conference. I'm Bill Skutch, and I'm especially pleased to introduce today's speakers. First, because I was able to postpone my scheduled retirement last summer and continue to work with our fantastic team these past several months. And second, because there are so many incredible advancements in CF research and drug discovery to talk about and share with you today. Today's plenary talk will highlight recent progress on the path to a cure. We announced the path 2 years ago as our largest initiative ever, with an initial target commitment of $500 million and the ambitious goal of treating the underlying defect in every person with CF and ultimately delivering a cure for all. We are thrilled with the amazing progress that has been made to accelerate treatments for individuals who are still waiting for a breakthrough. We have already evaluated more than 70 industry programs and finalized 18 new contracts since starting the initiative, convincing these companies to work on new ways to treat CF and overcome rare mutations in people that will not benefit from CFTR modulators. When we started, we knew this was going to be hard. We would not only need to apply breakthrough technologies in gene delivery and gene editing, but we would also need to discover new science and approaches that did not even exist. Today, you will hear about mRNA, gene delivery, gene editing, and stem cell programs, some of which will soon be in clinical trials, and others that are laying the groundwork to prepare for future therapies. In the last 24 months, we have also changed the way we do business, working directly with venture capital groups to draw the best scientific and drug development minds into CF. Our collaboration with the Longwood Fund was started just a year ago and has already helped create an entirely new gene editing company. A second company developing a novel form of CFTR gene delivery is also under development. But I'm particularly excited about another program that we just finalized with Flagship Pioneering, one of the most successful biotech venture groups in the world. This program is really unique. After nearly 2 decades of planning, the CF Foundation and Pioneering Medicines, which was created by Flagship, will bring together 3 separate companies in a novel collaboration where each company will provide a piece of the puzzle. What's really amazing is that these companies were not initially working on CF, but by bringing them together, we can supercharge their technologies, specifically around CF treatments. We're incredibly excited about this approach and think it could be a model for future programs where the CF Foundation leverages its resources to foster unique cooperation between different groups around a common goal, something we really need in the field of CF genetic therapies. We also know that our success absolutely depends on a large number of people working together across the entire community to provide a path forward for developing badly needed CF treatments. In many ways, our own laboratory, the CF Foundation Lab in Lexington, Massachusetts, plays a key role in this process, providing assistance, expertise, guidance, and reagents with the sole goal of accelerating the pace of progress and ensuring the success of others. So I'm very excited that today we will hear from 3 lab members about how we are working to assist scientists, companies, and venture groups around the world to pave the way for the path to a cure. Our first speaker will be Dr. Martin Mense, who has done a fantastic job in starting and leading the lab for the past 12 years. He will initially provide a brief overview on the phenotyping efforts to increase Trikafta access for people with rare mutations, and then an update on efforts to find new treatments for nonsense mutations. Next, we'll hear from Dr. Hilary Valley, who joined the lab 4 years ago and leads the gene editing team. Hilary will discuss some incredibly exciting advances in gene editing, including a whole new way to treat essentially all people with CF through a single approach. And then Dr. Jed Mahoney will discuss some very recent discoveries that have helped us understand the specific types of airway cells that genetic therapies need to target in order to be effective. Jed also joined the lab 4 years ago, and he leads the stem cell group. And finally, Martin will come back and finish off by telling us how we are pushing forward to take advantage of new advances in gene delivery and how we will use these strategies for delivering the next generation of therapies to people with CF. I'd now like to welcome our first speaker, Dr. Martin Mense.
Thank you so much, Bill, for that introduction. Bill got away easy with this few minutes. I have to now do a lot more work here. But hello and welcome everyone to the second plenary presentation. My colleagues Hilary Valley and Jed Mahoney and I are excited really to be here and feel very privileged to speak to you today about the progress in the path to a cure. We've been working toward and dreaming about a cure for CF for many years, decades really, and we all know that our dream is not yet a reality. Nevertheless, there are many reasons for optimism. We have a talented community focused on the cure. The cure for all people with CF cannot be achieved in isolation by an individual or small team. It requires a critical mass of scientists, new ideas, approaches, and experiments. While we have definitely seen important scientific progress since the Path to a Cure initiative was announced 2 years ago, I'm most excited about the momentum that we've been able to build. Since the last North American conference, we have gone from funding 15 company programs to 25. Additional programs are under negotiation, and we have partnered with venture capital groups to help launch companies with promising technology. We're not focusing on any single approach or technology and are committed to leaving no stone unturned. You may wonder, how is it even possible that so many companies and even many academic labs are willing to join the effort? This momentum is really a testament to the infrastructure and resources that the CF Foundation has built, including a patient registry that is the envy of most other diseases, the therapeutic development and clinical trials network, as well as the financial strengths and amazing community strongly committed to supporting the necessary scientific work. Together, these strengths make the foundation a very attractive partner. Last but not least, there's also the Foundation's lab, which serves as a resource and hub for CF science, training, and tools. Today, we hope to convey our excitement about the momentum on the path to a cure, highlight the scientific challenges ahead of us, share a few examples on how new technologies can possibly address those challenges, and also give you a sense for how our lab contributes to the overall effort. Hillary, Jed, and I have nothing to disclose. The CF Foundation's Path to a Cure initiative is the umbrella for all company programs and academic research projects that aim to bring new and better treatments and ultimately a cure to all people with CF. Let's start our journey together with the refresher on the main components to the path. On the left-hand side, in the yellow big circle, you can see it all starts with the mutated CFTR gene. From there, you can imagine this like a 3-lane autobahn. When I'm back in Germany, I prefer driving in the far left lane that speeds upwards of 100 miles an hour. The equivalent of this fast lane in the graphic is the top row, repair CFTR protein, which encompasses CFTR modulator therapies. We have been able to advance this approach the fastest, and it is the furthest along. In the middle lane, we are looking to restore the CFTR protein for mutations where modulator therapy alone is not sufficient, but we would either need to read through nonsense mutations or deliver CFTR mRNA, or in some cases, therapies with small oligonucleotides could bring the desired benefit. At the bottom with the yellow outline, we have the slowest lane, in which we look to fix or replace the CFTR gene through gene editing or gene transfer. Scientifically, this is the most challenging lane, and we've been able to pick up speed even in this lane in the last 2 years. Success here would eventually mean that we can reduce the 3-lane autobahn to just 1 lane, with all efforts going at the same high speed. Today, we will take a quick look at how we are doing in the fast lane, but our focus will be on strategies and approaches for the lower 2 lanes. As you will see, there are clearly distinct challenges for mRNA compared with gene editing therapeutics. At the same time, when it comes to delivering these kind of therapeutics, there are challenges common to both approaches. Let us begin by looking at CFTR modulators. Trikafta has been a life changer for those who benefit from it. This past June, the FDA approved Trikafta for children with eligible genotypes ages 6 and older. The clinical trial in younger children ages 2 to 5 is ongoing. Tomorrow's plenary session will cover the entire CFTR modulator landscape in greater detail, and in particular highlight the ongoing observational studies Promise and Simplify. Separately, work continues to make Trikafta available to more people with CF. Just before Christmas last year, the FDA added 177 rare and very rare mutations to the label, making an additional 600 individuals eligible for Trikafta, in addition to those who were already eligible because they have at least 1 copy of the F5 mutation. And we are not done. I'm really excited. In our research recently, we found that the more mutations we look at, the more mutations we identify as responsive to Trikafta. The CF Foundation has sponsored a serotyping study that was conducted in 3 labs and examined about 650 mutations, which also included those studied by Vertex. Vertex had found that more than 70% of the missense mutations they studied are responsive to Trikafta. And that essentially holds true in our largest study as well, which soon will be submitted for peer review and publication. A little over half of the mutations that we studied had such low baseline function that we would predict severe CF disease. In our lab studies, more than 2/3 of these mutations responded to Trikafta to a degree that we would expect to result in clinical benefit. These results are impressive, but it must be pointed out that the data from the CF Foundation study will not immediately result in further label expansion. We're sharing our data with Vertex, but only Vertex can apply for additional mutations to be added to the FDA label. Back to our path to a cure. Now let's move in the middle lane and look at nonsense mutations. Here's a quick overview of what nonsense mutations are. At the bottom, you see normal CFTR mRNA, which is the blueprint that machinery inside our cells translates into the CFTR protein. The mRNA has a green go or start signal where the translation machinery begins its work, and then a stop signal indicating that the full CFTR protein has been made. A nonsense mutation introduces a premature stop sign into the mRNA, which means the translation machinery stops its work early, resulting in a truncated, non-functional CFTR protein. As if this were not bad enough, cells have a control mechanism called nonsense-mediated mRNA decay, or short NMD. Through this mechanism, here depicted as a character from the classic Pac-Man video game, much of the mRNA containing nonsense mutations is degraded. Unfortunately, this means that our read-through therapies have to make enough full-length CFTR protein from fewer blueprints. Grouped together, nonsense mutations make up for the largest class of mutations with no effective therapy to address them. The CF Foundation is highly committed to developing pharmacological read-through therapeutics, and the actively funded programs bear testament to that. Icogen on the one hand, and the Southern Research, in partnership with the University of Alabama, Birmingham on the other, conducted large high-throughput screens and small molecule collections to identify possible starting points for new drug development. Both programs are now in the process of chemically optimizing hit molecules identified in the screening efforts. Elox Pharmaceuticals has a more advanced program and is currently testing a new medicine under the codename ELX02 in individuals with CF with nonsense mutations. We don't know yet how well the medicines work, but the good news is that it appears to be safe at the tested doses. At our lab, we're carrying out additional novel high-throughput screens, but we also collaborate with these 3 foundation-supported programs by exchanging liaisons and expertise. We may send gene-edited cell lines that we have generated to the companies, while the companies send molecules for testing in experimental models that we run at our lab. Over time, it has become increasingly clear that developing a medicine for nonsense mutations is an extremely difficult nut to crack. Currently, it seems unlikely that a single molecule can be strong enough to bring clinical benefit to all who have CFTR nonsense mutations. Instead, we may need to combine molecules to achieve the desired benefit, much like Trikafta is a medicine that contains 3 different CFTR modulators. The collaborations illustrated on the left may enable us to build a successful combination incorporating molecules from these 3 efforts. An example for how this could work comes from our lab. I was really blown away when we first saw this data at an internal meeting a few months ago. In the blue box on this image, the dark upper band, labeled band C, is a signal for 20% of normal CFTR. Now look at the red box. CFTR R11C22X is one of the most common nonsense mutations. When cells that express the CFTR mutant are treated with ELX02 together with another compound, the amount of the mature full-length CFTR protein exceeds 20% of normal CFTR. That is the same as what Trikafta achieves for CFTR protein with the F508del mutation. In the yellow box, you can see that each of the molecules on its own yielded little to no full-length protein. The power of combinations is very exciting to see. But remember, this is an early but very promising lab result, just illustrating what may be possible. As you're all aware, of course, small molecule therapeutics are not the only approach the CF Foundation supports, which may one day result in effective therapy for those with CFTR nonsense mutations. Let's look again at our path to a cure graphic and focus on the lower 2 lanes. Mutation-agnostic approaches, which encompass mRNA, gene therapy, and gene editing, aim to bring long-awaited improved outcomes to all people with CF who have no therapies available to them. With that, I'm happy to introduce my colleague Hilary Valley. She will explain how the various genetic therapeutic approaches are distinct from each other and then focus on recent advances and current ideas in gene editing.
Thank you, Martin. Hello, everyone. I'm thrilled to be here with all of you online. In this part of the plenary, we're going to dig deeper into those bottom 2 lanes of the path to a cure and I'm excited to share the momentum that is building in these areas. Before we dive into the details of gene editing, let's review how genes become proteins and therefore how modifying genes can result in modified proteins with improved function. A gene is a unit of DNA found in the human genome. Genes are made up of exons and introns. Exons contain the instructions for creating proteins, such as CFTR. When a cell makes a protein, DNA is first transcribed into RNA, which contains both exons and introns. The introns are then removed in a process known as splicing to create an mRNA transcript. The mRNA is translated by the ribosome to create a protein. CFTR is a large and complex gene. CFTR consists of 27 exons and 26 introns. After the introns are removed, the exons remaining in the mRNA transcript are over 4,400 nucleotides. For perspective, the average gene in the human genome has 12 exons compared to CFTR's 27, and the CFTR mRNA is almost twice the length of that of an average gene. As many of you are well aware, CF is caused by mutations in the CFTR gene, which results in a poorly functional or absent CFTR protein. In the CF population collectively, there are hundreds of mutations that can cause CF, and these mutations are found all throughout the CFTR gene. Most people with CF have one of these mutations on each of their 2 copies of CFTR. This schematic shows the 27 exons of CFTR, shown here as gray bars, and the 26 introns of CFTR, shown here as a gray line. CFTR mutations occur mostly in exons, but also in introns. Let's zoom in on exon 12. Exon 12 is the location of the nonsense mutation G542X, the most common mutation for which there is currently not an approved therapy that treats the basic defect. There are many different CFTR mutations throughout exon 12, and each red line indicates a CF-causing mutation that has been found in people with CF. Here I featured 2 mutations in blue that highlight the point that most CFTR mutations are rare or ultra-rare. After F508del, G542X is the most common CFTR mutation in the US, but it is still a rare mutation, with only 1,416 people in the US with this mutation. Other mutations, such as L558S, are ultra-rare, and there are only 12 people in the US with this mutation. Another way to think about CFTR genetics is to pretend the CFTR protein is a car. Everyone with CF has a car that doesn't function. Just as there can be many reasons why a car may not function, there are many mutations that can cause CF. Some people with CF have no steering wheel in their car. Some people with CF have a broken transmission. Our shared goal is to make all the cars run smoothly. And there are 3 genetic therapy approaches that could help us do this. The first is mRNA therapy. Here, the mRNA molecules are shown as red squiggles. mRNA has been in the limelight this past year with the development of the COVID-19 vaccines, and a lot of data has been generated to show that mRNA therapy can be both safe and effective. mRNA therapy is like a car rental. It gets you from point A to point B for a few days or more, but provides temporary transportation. mRNA molecules transiently provide the cell instructions for creating a protein, such as CFTR. We are excited about mRNA therapy for CF since it could be an effective therapy, but it will need to be repeated, and it is not a cure. The next 2 types of genetic therapies, gene therapy and gene editing, involve DNA molecules rather than RNA molecules. Gene therapies for CF consist of a DNA sequence that codes for a functional CFTR protein. Gene therapy is like giving everyone a brand new car. The third is gene editing. Gene editing is the auto body repair shop. Gene editing-based therapies encompass all therapies where the faulty CFTR gene in a person with CF is modified in some way. In the diagram, the original DNA is shown in white, and the edit is depicted by a red star. Gene editing can be as small as changing a single base of DNA, such as repair of the G542X mutation, or it can be larger, such as inserting a superexon, which I'll talk more about in a few minutes. In the rest of this section of the plenary, we are going to be focusing specifically on gene editing technologies because we currently think this is the most promising approach for a cure. This is a simplified diagram of CFTR showing only a few of the 27 exons. Again, the exons are gray boxes and the introns are black lines. As we've discussed earlier, in the CF population collectively, there are many mutations that cause CF located all throughout the CFTR gene, shown here as red lines. Because there are so many CFTR mutations and they are located all throughout the gene, it is not feasible to build a huge gene editing body shop with the tools and technologies to fix them all individually. We are committed to our goal of a cure for everyone with CF. We will not leave anyone behind. Specifically for gene editing therapeutic approaches, this means we need to strategically design one-size-fits-all therapies. That is a single therapy that can be a cure for every person with CF, including those with rare or ultra-rare mutations. One way to do this is a gene editing approach known as superexons. Superexons are akin to replacing large pieces of the CFTR car, such as replacing the whole front of the car, instead of building the tools and technologies to separately replace the front tires, steering wheel, transmission, and battery. This diagram depicts a superexon consisting of CFTR exons 2 through 27, as well as a splice site and poly-A signal, which is a marker of where a gene ends that helps tell the cell where to stop making RNA. This superexon is being inserted into intron 1. In general, superexons can provide a correct CFTR coding sequence for all CF mutations that occur after the superexon insertion site. Therefore, they can provide benefit for all people with CF who have one or more of their mutations after that insertion site. This particular superexon would provide benefit for all people with CF who have one or more of their mutations after that insertion site in intron 1, which is 99.9% of all people in the US with CF. Superexons are appealing in part because they are inserted within the CFTR gene, and the scaffold of genome structure around CFTR is maintained. We are really focusing on gene editing in the native gene location because we think this is likely to be safer. By gene editing in the native gene location, we can be the most certain that CFTR will be expressed only in cells that should express CFTR. When the superexon is inserted, nothing is removed from the genome, only added. The original copy of CFTR is still present in a slightly shifted place in the genome, but it is not used because the superexon contains signals telling the cellular machinery to stop after the superexon. As a community, we need the superexon approach or something similar to fulfill the promise and hope of gene editing to cure CF. In some diseases, everyone with that disease has the same mutation. CF is not one of those diseases. The superexon approach is a strategic approach with the specific landscape of CFTR and the genetic diversity of people with CF in mind. Momentum is building for superexons. We at the CF Foundation Lab, Dr. Matthew Porteous' group at Stanford, Dr. Patrick Harrison's group at the University College Cork, Dr. Brian Davies' group at the UT Health Sciences Center at Houston, and other academic groups and companies have been working on superexon approaches using cells in the lab. Dr. Susan Burkett and Steve Rowe at the University of Alabama at Birmingham have inserted a superexon into a CF animal model. It is still early days for the superexon, but the data that is being generated is very encouraging. Superexons are one concept for gene editing repair, but there are many possibilities for superexons, both in terms of where the superexon is inserted and what DNA the superexon is composed of. Sometimes in science, researchers have a serendipitous breakthrough, like the discovery of penicillin. However, more often, progress comes from consistent effort and stepwise learning over time. One such early step was work carried out at the CF Foundation's lab. We inserted into intron 22 a small superexon construct consisting of exons 23 through 27. Using our car metaphor, this is like replacing the trunk of the car and the rear tires. This superexon could benefit approximately 15% of people with CF in the US, all people with CFTR mutations after the insertion site, which includes those with the more common W1282X and N1303K mutations, as well as many others. This bar graph on the right shows CFTR function as a percent of normal CFTR function. In cells expressing normal CFTR, as shown in yellow, there was robust CFTR function. In cells expressing N1303K mutant CFTR, shown in black, there was little to no CFTR function as expected. When we inserted our first superexon into cells with mutant N1303K CFTR, CFTR function was partially restored by the superexon, as shown in light blue. We were quite surprised by this result, and it led us to try and figure out why this was happening and how to make it better. Once we figured out what was happening, we were able to make a better superexon. This next superexon is the same as the first, except for that poly A signal DNA, which is that termination signal that helps tell the cell where to stop making RNA. This made a big difference in how effectively we could restore CFTR. Now the superexon was producing almost 80% of normal, healthy CFTR, well above the level needed to provide therapeutic benefit. From this work, we learned that DNA components can affect how well a superexon works, and in part, how effective a therapy might be for an individual. Let's look at one more superexon. Dr. Matthew Porteous' team published a study just this year where they inserted a very large superexon, almost the entire CFTR gene, into exon 1 in a creative two-step process. In our car metaphor, this is like replacing almost the entire car and leaving just the front bumper. In this study, they selected for cells that had the superexon inserted, and excitingly, they were able to see full rescue of CFTR function. This bar graph on the right shows CFTR function. CFTR function was low as expected in CF cells, shown in black, compared to healthy cells, shown in yellow. CF cells that had the superexon inserted had full functional rescue, as shown in dark blue. The superexon data that we just saw is very encouraging. We have made significant progress on superexons in just a few years, but most of these studies are being done in cells in a lab, and there are many hurdles still to making superexons a therapeutic reality. It's clear that superexons are larger and more complicated edits than repairing mutations one at a time. We may well see therapies for individual mutations before we see a superexon therapy, and that's actually good news, because we can build on those technologies and provide real benefit to real people along the way. At the same time, I want to emphasize that superexons are one of the ways that we could make CF stand for cure found. And there is both great excitement and great momentum for this approach. A year ago, we didn't have any companies working on superexon replacements. Now, the CF Foundation is funding several companies working on superexons for CF. Importantly, the foundation is supporting groups in other areas as well that are critically important for making superexons a reality. The first area is new editing technologies. There are advantages and limitations to all of the current editing technologies, and there are several groups, including Rewrite Therapeutics, who are working on very creative, novel editing technologies to help address some of these limitations. Current technologies are advancing and evolving. Companies like LifeEdit and Metagenome are looking for smaller gene editing enzymes because that will give us more delivery options. New and improved models. Models for evaluating gene editing safety and efficacy are critically important. The University of Iowa, Case Western Reserve University, and University of Alabama at Birmingham are all at the forefront of developing CF models. New and improved delivery technologies. Delivery is absolutely critical for successful gene editing therapies. These companies are all working on novel approaches for delivery. In the next section, we are going to be talking more about delivery. I'm excited to introduce my colleague, Jed Mahoney, who leads the stem cell biology team at the CF Foundation's lab. And he will be giving you an overview of the cell types in the lung specifically and how this information can help us with delivery strategies.
Thank you, Hillary, and good morning and good afternoon to all of you. Today, I'm going to change the focus from the intercellular level, where Hillary gave a nice view of altering the gene source and sequence, and zoom out, not yet all the way to the individual with CF, but to the context of the lung epithelium and the cellular landscape within it. I'm focusing on this topic not only because of a few recent landmark publications, but because it also highlights how the CF Foundation and our lab interfaces with the academic community to advance the understanding of the basic biology required along the path to a cure. Now, the lung, as I imagine you are all aware, is an extremely complex organ that has evolved over millions of years to exchange carbon dioxide for oxygen. What I doubt everyone thinks about is how the lung also functions similar to our skin. That is, as a barrier to keep the outside environment out, protecting the body, or well, us, against the environmental pathogens such as viruses and bacteria. And the lung is quite good at this barrier function. And it's this barrier function that we must either break down or somehow get around to deliver those transformative, potentially curative next generation therapies for people with CF. The lung epithelium is comprised of two different compartments, the alveoli, where the gas exchange occurs, and the conducting airway system. This conducting system is comprised of a branched network of tubes that get progressively smaller as air is delivered down to the alveoli. Here, debris and pathogens are captured in the mucus while the air is warmed and hydrated. And it is in this conducting airway where CF is most destructive. And therefore, we've focused our research efforts to increase the understanding of these cell types and their functions. So let's take a look together and see what the airways are made of. Now here, on the left, you can see a histological section stained with hematoxyliniacin. In other words, it's a very thin section of a healthy conducting airway stained and just visualized through a microscope. The deep purple spots, well, those are the nuclei. This is where the CFTR gene is located and where Hillary was describing altering that gene sequence. The lighter pink areas, those are the cell bodies, or the cytoplasm of the cells, where the CFTR mRNA can be found. And then right along the top is where the CFTR protein is located. Here, now you can see what we refer to, now on the right is a cartoon representation of the airways, and we're highlighting the different cell types. Here, you can see what we refer to as luminal cells, or cells that face inward towards the air of the airway tubes. And these cells exist in a few different flavors. There are secretory cells that secrete mucins and other antimicrobial and lubricating molecules. Ciliated cells, these are the mechanical cell of the airways, which have extrusions that point out of the top of them that beat in concert to lift and clear the lung of debris. And the new and exciting ionocyte, recently discovered in 2018, which we know expresses significant amounts of CFTR. But the last cell that we're going to focus on, that's the airway basal cell. And this is a personal favorite of mine because it's the stem or the progenitor cell of the conducting airway. Interestingly, evolution protected this airway basal cell by concealing them in a specialized niche or a home that's safe from the dangers of the external environment. And it was kind of like hiding money under a mattress. So these cells, they laid down along the bottom of the epithelium, protected by a layer of those specialized luminal cells. Unfortunately for us, this protected niche makes them very hard to target, even with the most state-of-the-art gene editing and gene delivery mechanisms. But it is here, protected from the world around them, that the airway basal cells simply lay in wait for cells to be lost to either injury or even simple aging. Then they activate and do their job to repair the airway by replacing the lost luminal cells in a process we call differentiation. Now, this process is fascinating because not only must the basal cells replace themselves, but they also must regenerate the luminal cells that were lost and do so in a way that maintains the critical distribution of the cell types to allow for the clearance, airway clearance, and barrier function. And it is because of this process of repair through differentiation that these cells were actively trying to better understand. Imagine if we correct the CFTR gene in these stem cells, it will carry the corrected gene forward naturally as it functions to repair even the smallest of injuries through regenerating those new ciliated or secretory or other luminal cell fates. This, in effect, could be a tissue-specific or organ-specific cure, and one we are working tirelessly to achieve. Well, it's true. Even years ago, we knew tons and tons about these cells, their functions, and their distribution that comprises the conducting airway system. But even today, we still do not know enough to develop the targeted next-generation therapies. But we're making progress. In fact, as we make progress along this path to a cure, we're starting to ask new and interesting questions questions with finer detail than ever before, which require new and inventive data sets to answer. Now, we know today that we need an understanding of the CF airways at the highest resolution possible. Data sets that are designed to be flexible and forward-looking, capable of answering many different questions, including obvious ones like what cells and at what much is CFTR expressed, but other more nuanced questions, including who is really the airway basal stem cell, and what about them can we exploit for therapeutic benefit in CF? Now, understanding these immense complexities that would be needed to be navigated, about 5 years ago, the CF Foundation came up with the idea to form an epithelial stem and progenitor consortium, or a dedicated group of expert scientists that were tasked to lay the groundwork or pave and map the path to a cure. This consortium, led by CF's own John Sheridan, is one that I see as a great example of how the CF Foundation is bringing leaders in academic sciences together and inspiring them to focus on the needs of our community. And it is this consortium, which is a team. It's comprised of investigators from many different topics, ranging from the airway and developmental biology to bioengineering to CF animal models and all the way to bioinformatics. This team, we meet multiple times a month and we discuss cutting-edge topics in CF airway science, share insight, reagents, and protocols, anything that can help move the science forward. But most importantly, we share a commitment to the CF community to advance the science necessary to develop the transformative therapies for all people with CF, regardless of their mutation. Today, I'm happy to say the Epithelial Stem and Progenitors Consortium is succeeding to motivate and empower therapeutic development with the necessary transformative basic research we need for the cure tomorrow. Now here, I'd like to highlight some of the consortium's work in generating a molecular atlas of the CF lung. This work was led by Brigitte Gompers from UCLA and Dr. Barry Strips from Cedars-Sinai's Medical Centers and the CF Foundation Lab. But as you'll see on the author list, it was a full team effort. Just this last May, the consortium's work to map both the CF and the healthy airways were published in the Journal of Nature Medicine. This major multi-center collaborative study is one that is already having broad impact on the CF research community, as this data is being shared with academic and industry partners alike, advancing the mission. For this study, the consortia acquired a very large number of samples, one of the largest samples of its kind to date. Here, 40 human lungs, approximately 20 from healthy donors and 20 acquired at the time of lung transplant for people with CF, were processed for single-cell RNA sequencing. And the results are here on the left. At first glance, I'll admit, it doesn't look much more than just a speckled, colorful cloud. But to me, it's mind-blowing. Each dot represents an individual airway cell, and underneath each dot is an unbiased list of the genes expressed and at what level. The structure or the shape of this cloud is defined by each dot's similarity to all other dots, which remember, are all individual cells from one of those 40 donor human lungs. Next, coupling our understanding of the airway and using cutting-edge computational approaches, we color-coded those dots, or remember, really individual cells, as we defined them to the known cell types and even classified a few novel cell states never before described. Putting this together was akin to seeing color TV or, a 4K screen for the very first time. The clarity and the resolution were astonishing. Right off the bat, we learned a lot. For example, there are subtle differences in the CF and the healthy airways, but overall, the distribution of those cell types remained intact. We also believe, but we will need more experimentation to confirm this, that we resolve the process of differentiation at a transcriptional level. As you can see on the left, the structure of this data both has diffuse clouds or nodes, and these represent the cell types. And then there are thinner lines that are linking these nodes together. These thinner lines, or the roads between the nodes, we believe represent cell differentiation, or the molecular transition from one cell type to another. This resolution of the CF airway biology would not be possible without these cutting-edge single-cell technologies and being used at the scale that was done for this study. And this data that's being used is going to answer many current questions, but it will also be useful in questions that emerge and evolve during the therapeutic developmental process. For example, let's consider some of the major questions I outlined above, which is, what cell types express CFTR? Remember, it's not enough to just put CFTR in the airways. We have to put it back where we found it. And with this data set, we can easily ask that question and see a quantitative return. Here, you can clearly see that the secretory cell population is the cell type that dominates CFTR expression. This is not to say that secretory cells express the most CFTR protein on an individual cell level, so we believe that to be the ionocyte. Instead, this says that secretory cells are the population that, due to its frequency or their abundance in the airway, house the majority of CFTR expressed, and therefore become a prime target for gene therapies. And we can even enhance the resolution. We can look at which subtype or flavor of these secretory cells should we target. And you can see here, there's an orange secretory cell population known to the airway biologists out there as a club cell that dominates the secretory cell type that expresses CFTR. We believe, and studies like these are critical in our approach to accurately target gene therapies to achieve the functional CFTR expression. And we must get it in the right pace if we want to see these potential gene therapies, medicines work at their best. But there are issues with targeting just the luminal cells or secretory cells directly. Specifically, how long will these cells take, or how long will these cells and their therapeutic agent persist in the lung? This question has many factors that impact it. But in general, we believe that this will be only a treatment and not an organ-specific cure, as the lifespan of these luminal cells can be quite short. So this type of therapy would require repeat dosing. But if we got it right, and we got the right cells at the right dose, it could be transformative for the CF community. But we're not done there. We want the one-time cure. So how is this data helping advance the progress towards a cure? Remember how I described those nodes as the different cell types? Well, the lightest green node on the top left contains my favorite cell and our prime target for the cure in the CF lung, the basal stem cells. We're using this data to ask questions about what makes these airway basal stem cells different from all the other cells in the airways. And a mechanism that you'll hear more about in an upcoming video, we've started asking what receptors are specifically located on these basal cells that we can use as docking station for our therapeutics. I can say the answers that we're seeing today are being evaluated right now in the lab. So if we can target this cell with an appropriate gene editing strategy, like the Superexon approach that Hillary described earlier, we believe it will be a durable, long-term correction, a correction that is kept safe under the mattress. And these corrected basal airway stem cells, they will naturally and automatically replace those luminal cells lost throughout life with a cell containing a functioning version of the CFTR gene. In essence, an organ-specific cure. Now, to show you just one more example of how we will evaluate these types of delivery, I'd like to highlight the work of another epithelial stem and progenitor consortium member, John Engelhardt, and his lab at the University of Iowa. John and his colleagues have been hard at work developing the ferret CF model, a tractable and highly relevant model for CF lung disease. And understanding the CF therapeutic landscape and forecasting where we will need to go, John and his team have generated a ferret that can be used to assess delivery vectors, cellular targets in vivo. To do this, they generated a transgenic ferret, which has 2 fluorescent protein genes inserted in a very clever way. Under steady state, the entire animal will grow red due to the expression of TD tomato. However, if a gene editing event occurs, a recombination will switch the color from TD tomato, or red, to EGFP, or green. And so you can see here on the right, in the bigger picture, most of the cells are red in color, but the airways, which are sectioned across and look like circles instead of tubes, you can see green cells. These green cells are in the airway and indicate that a genetic recombination event has occurred and switched those fluorochromes. By generating data sets like these and novel animal models, the CF research community is paving the way towards the path to a cure. And we've just started. We've been busy building other data sets to generate and test hypotheses across the therapeutic spectrum. And as well, we're engineering the next animal models required to understand and optimize cellular targeting in vivo. And we're doing so much more that I wish I had time to share with you all today. But I hope one thing is clear. We're all in, and we will not rest until it's done. Thank you. And now I'll turn it back to Mark.
Thank you very much, Jed, for this great overview of the lung and the cell types lining the airways. Our knowledge on where genetic therapies would be most impactful has dramatically increased in recent years. To further illustrate the science and approaches for cell-specific delivery of genetic therapies, let's watch this video.
Genetic therapies for CF can be inhaled or given through an IV. However, getting these therapies into the correct cells in the lungs is not easy, and each of these delivery methods involves specific challenges. The lungs of people with CF contain a lot of mucus, which presents a barrier to all therapies that are delivered by inhalation. On the other hand, therapies that are delivered systemically through an IV must travel through layers of other tissues before reaching the correct cells. Once through these barriers, genetic therapies still need to target specific types of cells in order to have the desired impact. One way to do this is by using cell surface receptors. Receptors can be thought of as docking stations that only allow specific vehicles to park. The specialized vehicle that contains a genetic therapy, called a vector, must be able to dock onto a receptor so that the therapy can be taken up by the cell. Each cell type features distinct receptors on its surface, allowing us to target just the right cells for a given therapy. Messenger RNA and gene transfer therapies should be delivered only to cells that naturally express CFTR protein, like secretory cells and ionocytes. To prevent potential undesired effects, other cell types should be avoided. We can achieve this by targeting receptors that exist predominantly on secretory cells and ionocytes. For gene editing therapies to achieve lasting curative effects, they need to be delivered to the stem cells of the airway, called basal cells. Gene editing therapies are not expected to have undesired effects in other cells. In this case, we may be able to target a universally expressed receptor as long as it is present on basal cells. One additional challenge is that the cell receptors that face toward the blood vessels are different from those that face the airway surface. Delivering genetic therapies through an IV may require targeting different receptors than delivering through inhalation, even though the target cells are the same. While tissue or cell-specific delivery of gene therapies remains a challenge, together, we are making incredible progress along the path to a cure. Each step brings us closer to making these therapies a reality for all people with CF, and we will not stop until CF stands for Cure Found.
I love this video and the promising outlook it presents. But there was a lot of underlying complicated science. Let's consider one other analogy to illustrate the delivery problem before we recap the key messages. The Star Wars movies are popular in my family, and I'm certain many of you will recognize how they illuminate our work. Imagine a cell membrane through which the genetic therapy needs to be delivered is a protective shield around the planet. Then imagine the delivery vector is a cargo spaceship like the one you see in the left-hand image. The receptor on the cell is akin to a gateway in the shield, and only a spaceship that has the correct passage code can enter the shield and deliver its cargo. Just like on the right, you can see biological vectors being taken up by cells and delivering their cargo. Now back to the key messages of the movie. The challenges of achieving successful therapeutic delivery are not exactly the same depending on cargo and delivery technology. To avoid possible untoward effects in other cells and tissues, we think that mRNA or DNA for gene replacement should ideally only be delivered to cells that naturally express CFTR. Most of the CFTR exists in the secretory cells in red and the ionocytes in pink. Delivering these types of therapies will ideally be done with highly selective vectors. On the other end, for lasting curative gene repair, gene editing machinery must be delivered to what we just heard are Jed's favorite cells, the basal cells of the lung, from which the secretory cells and ionocytes are regulatory removed. No matter the cell or the cargo, a common theme is that in most cases, uptake will be mediated through receptors on the cell. On this slide, the receptors are the small stars and the vectors delivering the cargo are the larger stars. For inhaled delivery of mRNA or gene therapy, one may target a receptor that is only expressed on the desired target cells, the ionocytes or secretory cells. If the same therapies were delivered via the blood, the receptors or vectors would likely to be different. Here now in yellow, but the target cells remain the same. On the other hand, a curative gene editing therapy would need to be delivered to the basal cells, regardless of an inhaled or systemic delivery route. However, we don't expect any negative consequence. More likely it would be beneficial if gene editing therapy were delivered to as many cells as possible. The big question then is, how do we find the needed vectors? If you keep with our spaceship analogy, first, a suitable vector must have a cargo bay large enough for the therapy to be delivered. Second, the vector must be able to reach and be taken up by its target cells. And third, like a pill that one may take daily or weekly, the vector should be redosable. This is because a single dose of CFTR mRNA will probably not provide benefit for much longer than a week, and delivering the CFTR gene to the cells lining the lung airway would likely need to be repeated every few months because the cells on the surface die over time and are replaced with new cells. Technologies that are used or being explored for the delivery of genetic therapies are viral vectors and lipid nanoparticles or LMPs. And there's a third category that we have not covered previously at the plenary session, naturally occurring vesicles. These are kind of lipid nanoparticles, but they are normally secreted by most cells, including by those in our own bodies. We will review all these three categories, their use, strengths, and weaknesses in the next few slides. At the same time, we will hear about 4 examples where the CF Foundation has formed partnerships to explore delivery technologies that seek to improve on shortcomings of the more established approaches. Just to be clear, by shortcomings, I don't mean to suggest that an approved therapy using a certain vector is bad, but the properties of the vector used for an ophthalmology disorder, for example, may not be ideal for specific needs in CF. Here are more details on viral vectors. The most explored and commonly used viral vectors for gene therapy are the family of adenovirus-associated viruses, or AAVs. They occur naturally, don't cause disease in humans, and are used in 2 approved therapies. Those of you following gene therapies closely may have heard that the FDA held a summit to discuss side effects that have been observed in the approved therapies. No matter what will ultimately come of this increased scrutiny, a possible liability for AAV-based therapies is that the viral vectors are quickly neutralized by our immune system. That's not ideal for repeat dosing. We are excited to share that through our partnership with Longwood, we are helping to launch a new company with the goal to develop a different class of viruses for genetic therapies. We expect the lead program to be the development of human Boca virus for therapy in CF. The Boca virus has a natural specificity for lung, has cargo capacity large enough for the full CFTR gene, and can be dosed multiple times. Much of the CFTR-related work so far has been carried out by Dr. John Engelhardt's group at the University of Iowa. His team's results suggest that this virus can be a promising alternative to AAV vectors. But we certainly don't continue to believe that AAV vectors can play a major role in CF therapies. The CF Foundation supported AAV programs at 4D Molecular Therapeutics and Spirovant continue to advance toward clinical trials. And we also have a new partner in the AAV space, Splice Bio, which has found a way to improve on tissue and cell specificity of AAV vectors by decorating them with proteins that home the viral vectors to target cells. This is achieved through intim protein domains that are spliced out, resulting in AAV vectors now only decorated with the target-specific proteins. Viruses can be decorated with different targeting proteins, allowing them to selectively bind to cells with receptors matching those targeting proteins. This technology has not progressed past laboratory testing yet, but promises unprecedented target-specific uptake for AAV vectors. Probably most of you have heard about LMPs, or lipid nanoparticles, because they are used to formulate the COVID mRNA vaccines from Pfizer and Moderna. A major challenge for this technology is how to make them selective for just the target organs and cells, in particular, if one were to deliver via the blood to the whole body. Dr. Dan Sigwitz's group at the University of Texas Southwestern Medical Center has made impressive progress towards organ selectivity after intravenous injection. starting with an LMP that can target the human liver, they added lipids with specific properties and found that depending on the amount and kind of lipid, they could target the LMPs to the lung or the spleen instead of the liver. Again, this technology has not been tested outside the laboratory test setting yet, but it appears to be a major advance for the use of LMPs in CF and target delivery to the lung. The publications in the last year and a half in premier scientific journals emphasize the great importance of this technology, which has been licensed to Recode Therapeutics, a partner in the CF Foundation's path to a cure. Natural lipid vesicles are the new kids on the block among delivery technologies that are being explored for the use in CF. They are naturally secreted by most cells in our bodies, and depending on the exact form of secretion, they may be called either exosomes or extracellular vesicles. Because these vesicles are naturally occurring as vectors, they are deemed safe for humans, but that has not been confirmed in any therapy yet. Depending on the cell that produces these vesicles, they may naturally have the right targeting proteins on their surface, or one might engineer the producing cells as needed. Also, cargo for genetic therapies could be generated and loaded by the producing cells. Alternatively, these vesicles could be harvested and then loaded by physical means. Emblematic of the interest in the naturally occurring vesicles, there are over 30 companies working to harness the potential of this technology. The CF Foundation has a partnership with a group of Dr. Matthew Wood at the University of Oxford in England, and a new company, Carmine Therapeutics, which generates extracellular vesicles from O- red blood cells. O- is a universal blood donor type, so that these vesicles can potentially be given to all people. Recognizing the ongoing need for effective and targeted delivery for CF genetic therapies, we are thrilled to be exploring extracellular vesicles as yet another potential option. With this, we have reached the end of our technology review. Here's a full summary of the CF Foundation's PTAC initiative. We are actively supporting a total of 25 programs across the areas of CFTR restoration, gene delivery, gene editing, and so forth. More programs may be announced soon, and we have entered partnerships with venture capital groups to create new companies. Our different venture partners are diversified with respect to how they source scientific ideas and technologies, and also with respect to their different geographical footprints. We are very happy that Flagship Ventures is our newest partner in this space. You may not be aware that one of the companies created by Flagship is Moderna. Since our last NACFC, we have had 153 meetings with companies, established partners as well as potential new partners, entered 10 new partnerships, and more will be added soon. We don't have a crystal ball and cannot see when exactly we will have approved genetic therapies, but we are extremely encouraged that we have been able to attract so many partners. I'm sure we all feel the excitement and promise from having built such momentum and having so many interplanetary explorers, just to put it one more time into Star Wars terms. Along with sharing our enthusiasm over the ongoing science and programs on the path to a cure, it was our goal to give you a sense of the central role played by the CF Foundation's lab in Lexington in supporting our mission of finding a cure for CF. For this section on delivery, you did not hear specifically about the role of the lab, but rest assured that we interact with pretty much all the companies in the delivery space. I would like to acknowledge the team working at our lab. It is a sign of the times that you recognize this as a Zoom team meeting photo, but we have been operating nonstop since the middle of June last year. This very last slide is meant to give you a glimpse of our lab's worldwide interactions. The yellow marker in the northeast of the US indicates the location of our lab just outside Boston. The white lines show you to where we have distributed cells. The yellow lines go to university labs that we interact with. And the blue lines go to locations of companies that we've helped or worked with. Some lines represent several partners. And the zoom out shows you that our interactions reach much further than just North America. This animation clearly demonstrates the global reach of the CF Foundation's research effort, as well as the commitment by companies and academic institutions to crack the code for CF genetic therapies and move us down the path to our ultimate shared goal, a cure for CF. This brings us to our journey together through progress, tantalizing scientific excitement, and promise to the end for today. Hilary, Jed, and I would like to thank you very much for listening to us.